Hi everyone! Today we're going to be talking about T cell subsets and functions. Essentially, we'll be talking about what happens when a T cell becomes activated. This is a continuation from our last lecture, so you may remember that although all the figures in this presentation are from the textbooks, you may want to pay attention to the bottom of the screen where these figure numbers will be in white. You can then follow along in your textbook and see the images if they're not as clear on the presentation. Today, we're going to discuss the two major subsets of T cells, cytotoxic CD8 positive T cells and helper CD4 positive T cells. We'll then learn about the roles of the major CD4 positive helper T cell subsets and how they're induced. We'll also learn about the effector functions of these different subsets of helper T cells. And finally, we'll talk about how cytotoxic T cells can recognize and kill a target cell. We'll first start off with understanding how the two major subsets of T cells, CD4 positive and CD8 positive T cells, differ and how they're induced. You may remember from previous lectures that the CD4 and the CD8 T cells are decided during the development phase when T cells are still in the thymus. This is dependent on whether T cells see MHC class 1 or MHC class 2 when they're in the thymus. You'll recall that MHC class 2 can only interact with CD4 positive T cells, while MHC class 1 will only interact with CD8 positive T cells. This is highly important because the CD4 positive T cells and CD8 positive T cells act very differently. Who they see in the thymus then determines what their function will be for the rest of their life. What's important to see on this slide is that CD4 T cells, as we remember, are activated by MHC class 2. However, they also receive additional signals that will then cause them to express CD40 and CD40 ligand on their surface. This allows them to interact with the antigen-presenting cell, which will then further the effector function of the antigen-presenting cell. And we also know that CD8 positive cells from the previous lecture are activated by MHC class 1, and this, as we'll see, is further enhanced by the CD40 and CD40 ligand interaction between the CD4 positive cell and the antigen presenting cell. Now we know that CD8 positive cells can be activated in the canonical 3-signal step pathway we remember from the previous lecture. Recall that first, a T cell needs to see its antigen on the MHC. Then, it needs to receive co-stimulation from the antigen-presenting cell via B7-1, B7-2, signaling through their receptor CD28. And finally, the T cell needs to receive cytokine signals from the antigen-presenting cell that will tell it what kind of cell to become. However, CD8 T cells sometimes need a little extra help. Sometimes, when the antigen presented to the CD8 only weakly interacts with the CD8 T cell, it needs help from the CD4 positive T cells. When CD4 positive T cells interact with the APC, they cause the upregulation of certain co-stimulatory molecules like CD40 on the antigen presenting cell, as well as 4-1BB. This is one of the co-stimulatory molecules we talked about last time. Note that unlike CTLA-4, instead of inhibiting the activation of the T cells, it actually drives the activation of the T cells. Additionally, activated CD4 positive T cells will also secrete IL-2. IL-2 is an important cytokine we'll discuss in later lectures, but for now, what you need to know is that IL-2 is incredibly important for driving the proliferation of T cells after they've become activated. So, when CD4 positive T cells interact with the APC, they can upregulate IL-2 expression as well as 4-1BB and CD40. This allows for the CD8 positive T cell that's interacting weakly with its antigen presented on MHC class 2 to get even further activation signals. This will allow it to overcome a threshold and then become activated. The CD4 T cell further confirms to the immune system that this is an antigen we should be responding to, and thus helps the activation of the CD8 T cells. So what are some of the specific roles of the CD8 versus a CD4 positive T cell? Well, first let's start with the CD8 positive T cell. CD8 positive T cells are also referred to as cytotoxic lymphocytes. This can be broken down into its roots: cyto meaning cell, toxic meaning poisonous. Essentially, a CD8 positive T cell is poisonous to its target cell, which will ultimately result in that target cell's death. We also know that CD8 positive cytotoxic T cells will recognize virally infected cells or cells that have been infected with an intracellular bacteria. We know this because MHC class I, which presents the antigen to CD8, only picks up peptides from inside the cell itself. Recognition of the antigen presented on MHC class I by the infected cell triggers the CD8 T cell to kill that target cell. Effector molecules released by the CD8 T cell, these are many different cytokines we'll talk briefly about later, then kills that target cell. So you can see here the CTL, which stands for cytotoxic lymphocyte, will recognize a virally infected cell and then kill the cell using effector molecules. That cell dies via apoptosis, which is a non-inflammatory cell death, and thus the virus no longer has a host cell in which to replicate. So, if we start at the beginning of an immune response, a CD8 positive T cell will become activated via the 3-signal step, or will become activated with the help of the CD4 T cell that has already been activated. It will then undergo a round of proliferation driven by the presence of the cytokine IL-2. It will then differentiate into the CD8 positive T cells via additional cytokine signals, after which it can then survey the site of infection, identifying infected cells and killing them via effector molecules that will lead to the apoptosis of the infected cell. So what about CD4 helper T cells? Well, CD4 helper T cells, as we've learned, can also help the CD8 positive cytotoxic T cells. They also, in addition to providing activating signals to the T cell, can provide activating signals to the B cells as well, and will help them determine their antigen specificity as well as the isotype that they will adopt later on during the infection. Additionally, release of specific cytokines from the helper T cells will help the immune response tailor the way that it responds to the infection based on what it's infected with. So these are a lot of different functions that the CD4 helper T cell has. How does it perform all of these functions? Well, that leads us to our next objective for the day, which is to learn the roles of the major CD4 helper T cell subsets and how they're induced. So unlike the CD8 positive T cells, there are actually a whole slew of subsets for CD4 positive T cells. This provides specificity to their function and allows them to act in a manner that is specialized towards the type of infection that the body is experiencing. There are many different subsets, including Th1, Th2, Th17, T follicular helper cells, and T regulatory cells. We're going to go through each one of these in the following slides. But before I begin, let's first understand that in order for these subsets to be induced, they need to receive certain cytokine signaling from the antigen-presenting cell. You may recall that this is actually signal 3 of the 3-signal activation steps, which then tell the CD4 T cell what kind of helper it needs to become. Once those T cells have differentiated into their specific subset, they can then produce another set of very specific cytokines that will do different functions depending on the type of infection the body is experiencing. Those cytokines will activate other cells, recruit other cells, and tell the body how it needs to respond to deal with the infection it has appropriately. So let's start with Th1. Th1 cells are very important for responding to intracellular bacterial infections and some viral infections. This allows them to help the activation of the CD8 positive cytotoxic T cells. This is important, as you may recall, because the CD8 positive T cells are very important for responding to viruses and intracellular bacteria due to the specificity of the MHC class I. Th1 cells also recognize macrophages carrying bacteria and activate them to kill the bacteria via phagocytosis. All in all, the functions of the Th1 support what we call a type 1 immune response. This is something that we'll address a little earlier, but keep this in mind. An immune response that's characterized by Th1 differentiation and effector function is an example of type 1 inflammation or a type 1 immune response. The second type of subset we'll see today are Th2 subsets of helper T cells. Unlike Th1 helper T cells, Th2 are good for responding to helminths and parasites. They promote the response of helminth and parasite fighting by activating eosinophils, mast cells, and the IgE-producing B cells. When the Th2 cells become activated, they induce what we call a type 2 immune response. Before I go on, I'd first like to mention that a type 1 and a type 2 immune response are vastly different in their effector function. Because of this, a type 1 immune response is not appropriate to respond to a type 2 infection, and a type 2 immune response will not effectively deal with a type 1 infection, such as an intracellular bacteria or a virus. A good example of this are people who experience or suffer from tuberculoid leprosy. Some patients will activate a type 1 response, and some patients will activate a type 2 response. However, these patients respond very differently depending on what type of immune response their body has mounted. You can see that, I'm going to start over on this slide. A good example of type 1 and type 2 immunity not being able to act at the same time is during a Mycobacterium leprae infection. Some patients that are infected with this bacteria will mount a type 1 response, and some will mount a type 2 response. However, these patients act differently depending on what type of immune response they've mounted. Many of them will not respond well, while others will respond well if they've activated a different type of immune response. So you can see that patients who are infected with this bacteria who mount a type 1 response develop a disease called tuberculoid leprosy. You can see here on this side of the graph that in those patients, the organisms or the bacteria are present at low levels. They have low infectivity in the host. There's granulomas and local inflammation, which we'll talk a little bit about later, as well as peripheral nerve damage. But there's normal serum Ig levels and normal T cell responsiveness. So these patients typically do well when they're infected with the bacteria. However, patients who mistakenly activate a type 2 response, something that is more suited for responding to helminths or parasites, develop a disease called lepromatous leprosy and cannot control the infection as well as the patients who mount the appropriate type 1 response. These patients have organisms or bacteria that show much growth in the macrophages. The bacteria is highly infectious in these patients. There's disseminated infection as well as extreme damage, not only in the nerves, but also in the bone and the cartilage. There is a presence of hypergammaglobulinemia, which basically means you have B cells that are secreting a lot of IgG to respond to the infection, even though it's not working well. And you also have low or absent T cell responsiveness, so you're not responding as well to the bacteria as you should. So you can see here that type 1 and type 2 immune responses are very different, and you must activate the appropriate one in order to respond well to what you're infected with. So let's move on. The next helper subset is a Th17 helper cell. Unlike Th1, which respond to intracellular bacteria, Th17 helper T cells respond to extracellular bacteria. They amplify the neutrophilic response and they activate epithelial barrier defenses. When helper T cell subsets of the Th17 subset are induced, they then mount what's called a type 3 immune response. And second to last, is the helper T cell subset, T follicular helper cells. Unlike the other T cell subsets, T follicular helper cells are uniquely evolved to help B cells and activate them, as well as mediate class switch recombination after the B cells have become activated. TFH cells are necessary for mounting a mature B cell response. Somatic hypermutation, which you will have learned about in earlier lectures, is dependent on the presence of T follicular helper cells. T follicular helper cells will select for B cells that respond very well to the antigen and allow those that respond less well after somatic hypermutation to be neglected, to die and not go out into the periphery to fight the infection. They're also important, as I mentioned, for coordinating isotype switching. So in order for the B cell to express different types of IG like IgM, IgD, IgA, IgE, etc., the T follicular helper cell needs to be there. Finally, unlike the other T cell subsets, T follicular helper cells are only found in germinal centers. These are the B cell centers of proliferation and affinity maturation that form when the body has been infected and B cells have been activated. Our final helper T cell subset for today are T-Regs. T-Regs are also different from the other helper T cell subsets because they don't activate or further encourage an immune response. In fact, they do the opposite. T-Regs are important for dampening the immune response. They inhibit the activation of dendritic cells and thus the activation of T cells. They can also be thymically derived or induced in the periphery. That means that if a self-reactive T cell is not deleted during development and it's found to be in circulation after the fact, these T cells can then be induced to become T regulatory cells. That means that if they see a self-antigen, they'll ensure that the immune response is not mounted as a result of recognizing self instead of a foreign pathogen. It's not, okay, there. As I mentioned earlier when we began talking about the different T cell subsets, all of this is determined by the third signal of the 3-signal step activation we talked about in the previous lecture. Remember that in the first step, T cells must see antigen presented on their appropriate MHC class 2. Then the T cells must receive a co-stimulatory activation signal from the antigen-presenting cell. And finally, they must see a set of cytokines that will inform the T cell what kind of subset it needs to become. These can be activated by many different types of cytokines, and we'll get into how the different cytokines induce the different types of T cell subsets now. So, as I've mentioned, the cytokines will specify the fate of the activated helper T cell. Remember that whether a T cell is Cd4 or CD8 has already been determined much earlier in development in the thymus. Once they're out in the periphery and become activated, cytokines will then tell them what kind of subset of helper T cell the Cd4 T cells need to become. CD8 T cells are already cytotoxic T cells and don't need the same type of specification. So first, the Th1 T cells that we talked about, the ones that mount an inflammatory type 1 response, will be activated by the cytokines interferon gamma and IL-12. These are produced by the antigen-presenting cell that has activated that T cell. Th2 cells, the cells that mediate a type 2 response to helminths and parasites, are activated by the cytokine IL-4. Th17 cells, which activate the type 3 response that respond to extracellular bacteria, are activated by 3 cytokines, TGF-beta, IL-6, and IL-23. T follicular helper cells, the ones that help the B cells, are activated by IL-6. And finally, T regulatory cells, the ones that are induced in the periphery after development, are activated by TGF-beta and IL-2. You can see here that many of these activating cytokines, the one on the top row here, are shared between subsets. However, the subset cannot be committed to unless all of the necessary activating signals are present. For example, IL-6 alone will always activate a T follicular helper cell. Only in the presence of IL-23 and TGF-beta can the Th17 cells be induced. The same goes for TGF-beta. Only when TGF-beta is with IL-2 can the induced T regulatory cells occur, and only when TGF-beta is with IL-6 and IL-23 can Th17 cells be induced. Once you have the activation of all these subsets, the subsets then produce their own unique set of cytokines that will then allow them to perform their specific effector functions. Th1 cells will produce the type 1 inflammatory interferon gamma. Th2 cells will release IL-4, IL-5, and IL-13. These are important not only for responding to parasites and helminths, but also for activating an allergic response. Th17 cells, true to their name, will produce IL-17 and IL-22, which help induce the neutrophilic response we talked about earlier, as well as activate epithelial barriers. T follicular helper cells will secrete IL-21, which helps with the development and maturation of the B cells after they've been activated. And T regulatory cells will produce the anti-inflammatory cytokines TGF-beta and IL-10. You'll hear more about these in a later lecture. So how do these T cells actually respond to the transcription factors that tell them what kind of subset they need to become? Well, the cytokine signaling that they receive drives the activation of specific transcription factors. These transcription factors will then go into the nucleus and drive the expression of a set of genes that will define the identity of that T cell. Certain transcription factors will only respond to specific cytokine signaling, which allows these subsets to be highly regulated and specific to the infection at the time. Interferon gamma, which we remember induces a Th1 response, induces the transcription factor STAT1. IL-12 induces the transcription factor STAT4. Together, the two of these drive the expression of Tbet, which will further characterize the Th1 subset. A similar thing happens with Th2 T cells. IL-4 drives the activation of STAT6, a transcription factor within the T cell. STAT6 will then drive the expression of GATA3, which then helps identify the Th2 by driving another program of genes. IL-6 and IL-23 together can induce STAT3, the transcription factor, which will then activate the expression of ROR gamma T. ROR gamma T then drives the expression of a set of genes that will define the identity of Th17 cells. IL-6 alone will also activate STAT3, but in this context, without IL-23, STAT3 will only drive the activation of BCL6. BCL6 then defines the TFH identity. Finally, IL-2 alone will activate the transcription factor STAT5, which drives the expression of FOXP3, which defines the T-Reg. These ones in the white boxes here, Tbet, GATA3, ROR gamma T, BCL6, and FOXP3, are highly definitive and are very good for identifying these different T cell subsets. That means that if I have a set of cells and I want to know what kind of subset they are, if I look at these markers, I can know whether they're a Th1, Th2, Th17, TFH, or T-Reg. These transcription factors also allow for the expression of those cytokines that we talked about earlier that perform the specific effector functions of the different subsets.